clear that you are the strongest attendance to the conference, and we all are in agreement to thanks to Jorge for the wonderful gala dinner yesterday, and with surprises and emotions along not only yesterday but along these days. So it was amazing, quite singular. So today is a big honor to present to first of all, a very good friend, and also to one of the most relevant and outstanding Spanish scientists with an international relevance, which is on the top of our scientists in our country. Let me tell you something about this important chemist we have. He studied at Santiago de Compostela University. It's one of the oldest universities in Spain, with a very good and old tradition, particularly in chemistry, in science, but particularly in chemistry. He was chemically educated there, but soon new universities were created in Spain, among them in Galicia, but in particular universities like La Coruña, Vigo, and this was a very good opportunity for young and talented scientists, and this was the case with Luis, who decided very soon to move to University of Vigo. And just 10 years later, he got the position as full professor in this university. That was in 2006. And well, in Vigo, that was a critical point in his career, scientific career, since he created really one outstanding research group, who was probably the responsible because 10 years ago, roughly 10 years ago, he was appointed with a position in Iker Basque, is the scientific system in the Basque Country in Spain. And well, the reason because of this appointment is because he was appointed as the scientific director of one of the most important institutes in the Basque Country, the CIC Biomagune, that he has been conducted for 1 decade. And he stopped very recently because he was eventually offered to come back to the Vigo chair, and now he has both positions in Biomagune and also Vigo University, where he has the chair in the physical chemistry department. And well, it is important to say that he has created a group, a very expert group in nanoscience, and particularly in achiral and chiral plasmonic nanoparticles, or nanomaterials in general, as today is the presentation. Let me give you, I said in my previous presentation about, I don't like to say numbers, only when I am really impressed with the numbers is when I like to say something about that. He has published over 600 papers, and over 500 papers, but he has got around 60,000 citations, and the index, age index in Spain is probably, in chemistry, should be among the first 3 scientists, with roughly around 130, which is only a few chemists are able to surpass the barrier of 100. So we are really in front of a really outstanding chemist in our country with an international projection. Because of this career, very successful career, he has received a lot of awards. Let me tell you that only one abroad, like for instance the Alexander von Humboldt Award, which is given by the President of the German Republic. And in Spain, he has received what could be many, but we could say that the National Award in Chemistry has also been received by Luis. And not only this career was reflected in these awards, but also in the editorial positions he has maintained along the many last years. In particular, he has been very much engaged with American Chemical Society journals. He started with Landwehr. He continued with ACS Omega in the foundation of this journal. And now he is executive editor in ACS Nano. So if you have really important papers to submit, Luis can help you, assuming that you pass the threshold of the quality. Huh? Okay, so it's my great pleasure to give the floor to Luis for his presentation that today will be focused, as you can see here, on plasmonic nanomaterials. So, please.
All right. Good morning, everybody. First of all, thank you for being here. As Nazario indicated, this is the most difficult session in any conference, the one after the conference dinner, in particular with a conference dinner as we had last night. So I would like to join the thanks to Jorge and all the other people helping with the organization. Now I understand why Nazario asked me to remain sitting while he was introducing me, because it took a while. But anyway, thank you for your warm words. Now I will have to take 5 more minutes because he used them. But, I'm going to start by a reflection that given the weather that we had this week, I can only offer you to come to San Sebastián, the sunny place. In fact, if you look at the tourist announcements, in Germany they call it the happiest city in the world. And I'm sure some people here will agree that there are many reasons for that. But honestly, this was not the only reason that I moved there. In fact, I like to show this other picture of the city where you can actually see the technology park, and this is where CIC Biomagune is based. So as Nazario indicated, we are the Center for Cooperative Research in Biomaterials, and the main focus of the center is related to bioimaging, theranostics, and what we call synthetic bioengineering, where we have actually one of the top setups for preclinical bioimaging. With the cyclotron, radio imaging cameras, and also MRI cameras, plus all the other equipment for characterization of materials and so on. So, in this center, I lead the so-called Bio-Nanoplasmonics Lab, which we could define as plasmonic nanomaterials for biomedicine. And here we started recently an ambitious research program related to what we call 4D surface-enhanced Raman scattering, biosensing, and bioimaging, which is funded by an ERC grant. And so the objective of this project is building 2 more models that will help us understand the dysregulated metabolism that takes place in cancer. And so the idea mainly is to build scaffolds that will contain plasmonic nanoparticles that should act as sensors to detect the metabolites related to this abnormal behavior in cancer tissue, and also to fabricate encoded nanoparticles with which we can label cells, so that in the end we can do double detection related to sensing with these particles and monitoring the cell distribution in 3D and over time, meaning in 4D, through the labels that we can attach to the different types of cell lines comprising the tumor. So this is, of course, a very ambitious program because we need to learn how to prepare bioprinted scaffolds, how to incubate the combination of different cell lines that are relevant to describe a tumor. And I will explain you a bit more in detail how we do things and why we do things. So first of all, we had already the opportunity to see wonderful lectures related to Raman scattering for detection. And so now you should all know that this is related to vibrational modes in molecules. And what is more important for what I'm going to tell you is that in standard Raman spectroscopy, you're exciting electrons typically to forbidden states, and then they are scattered by going down again to the same ground electronic state, but different vibrational state. So the emission of these photons has a very low probability. And this is why for doing standard Raman scattering, you need typically a rather large amount of material. Now, it was discovered a little bit by coincidence, as usually happens in science, that if molecules are located on top of a rough metallic surface, then the signal of the Raman scattering, the signal of the scatter light in this process, is enhanced by many orders of magnitude, typically scaling as the fourth power of the enhancement of the electric field at the metallic surface. And this is related to plasmonics. Okay, so when we radiate a very small metallic particle, we have a very well-defined absorption and scattering of light, but we also have a large enhancement of the electric field at the surface of the particle, which is important to enhance the Raman scattering process of molecules attached to it. If we want to optimize this value, we can either tune the morphology of the particles, or we can create assemblies. You remember the grapes from the theater. So exactly what we want to do is to organize particles in a way that they will concentrate the electric field at the gaps between the particles. So if the molecules that you want to detect are located here, this enhancement may be as high as to allow you to detect one single molecule. So the potential of this technique is huge, and this is why we have been working on it for a number of years now, and this is why we proposed that it could be used for this ambitious project. If you want to learn more about this technique, I would recommend that you follow this paper in this very important journal. This is, as you can see here through the list of authors, these are most of the main scientists working on surface-enhanced Raman in the world. And we joined efforts to publish this review article, which is almost 100 pages long and really contains most of the basics, the characterization, and the applications of surface-enhanced Raman. To be a bit clearer on what is this technique that we are using and how useful it can be, but also how difficult it can be, I have this list of advantages and drawbacks of surface-enhanced Raman. So it can be very selective because you typically measure the spectroscopic fingerprint of molecules, so you can really distinguish a molecule from all the others. It can be very sensitive. I mentioned already the possibility of single molecule detection. You can carry out very fast measurements. You can use portable instruments, so you can even take it to the field or to the clinic. Encoding capability, I mentioned briefly. I will tell you more about this. And it can be applied to almost everything because most of the molecules that we know are active in Raman scattering. The problem is, if you want to really have a controlled detection, you need reproducibility and uniformity in the fabrication of the plasmonic substrates that will help you enhance the signal. And this is usually not so easy. Additionally, in order to exploit the enhanced electric field, the molecules to be detected have to come very close, ideally, to the surface of the metallic nanoparticles. And so depending what type of systems you're measuring, in particular in biological systems, it's usually very difficult to avoid blocking the surface of the particles with biomolecules, like lipids, proteins, or other things floating around. And so you have to find ways to attract your analyte to the metal surface. And the same about contamination of the signal. Because of this attachment of other molecules present in the biofluids, you can actually get peaks that you don't want to see. Finally, this is something that is not really so much mentioned, but if you want to do measurements over time, there are memory effects that can actually make you confuse what you're measuring. So if analytes are absorbing at a certain time, they may not leave the substrate. So if you're measuring again, you may still see what happened in the previous time point. So these are obstacles that we have been trying to resolve for a number of years, and many PhDs and postdocs. So our expertise initially was on the colloid chemistry to fabricate metal particles with a high degree of detail, and also trying to understand the mechanisms involved in the growth of the same metal into different morphologies, in a way that we can control the dimensions and the morphology, and to obtain a very high degree of monodispersity in each system. And this is important because these morphologies, they all react with light of different wavelengths. And so in principle, this is an advantage because you can tune the position of the main absorption and scattering of light. But if you're not careful with what you're making, it may also be a drawback because you can get a very broad signal. Now, if you do it properly, then you can select particles that are responsive to light in the transparency window for tissue. And so you have seen already, I think, in Danny's talk at the theater and probably in many talks, this map of biological transparency windows. So we can actually tune the dimensions of these particles so that their plasmon resonances fall into any of these windows. And then you're set up to work with biological systems. However, for doing surface-enhanced Raman, what people typically do is taking a drop of any of those colloids, putting it on top of typically a glass slide, let it dry, and then say the molecules will fall into the hotspots, and we will get a signal that will tell us what we are trying to measure. However, if you try to make the most efficient surface-enhanced Raman substrate, you don't want something like this, where if you look carefully, so this is actually a computational simulation of the enhancement of the Raman signal of molecules located everywhere on this substrate, which is reproducing a typical deposition of gold nanorods on a substrate. So the white spots that you see here would be the places where the electric field is enhanced a lot, so you get a very high sensitivity for molecules falling there. But all the dark areas, or even the green areas, are places where the enhancement is very low. So just look here, what is the percentage of white spots that you get on this area. So you're wasting a lot of surface, you're wasting a lot of metal, so to say, because the signal is coming mainly from the white spots. If you change the morphology and you calculate again, you may even be in a worse situation. So gold nanostars, I showed you a plot before where the tips of nanostars would be very bright because they are known to accumulate electric field. So these are the places where you want your molecules to fall so that the Raman signal will be enhanced at the maximum. However, if you bring them very close to each other, you may even get destructive interference so that you get a lower signal than when they are separated from each other. And so this is what you see here. This would be the overall signal that you get from a distribution like this. Here you have a higher density of nanostars, and you get like a black hole in the middle, meaning that the signal will be decreased. And this is very important because it's very usual to see a spectrum of the colloid and then say, and now we put it on a surface and we measure with the wavelength at the maximum of the dispersion. This doesn't work because there is lots of plasmon coupling that will change completely your signal. So you really need to understand what you have directly where you're going to do your measurement. And so this is a work that we did together with Javier García de Abajo at ICFO and with José Manuel Taboada and Fernando Velleiro at University of Vigo and University of Extremadura, where we basically calculated the SERS enhancement for increasing coverage of spheres, rods, and stars. And what you see is that for spheres and rods, the signal is gradually increasing as you get more and more particles on the same area. And then there is a sharp increase when you start to have hotspots because they get very close to each other. But in nanostars, you can get a very constant signal because what you're gaining by putting more particles, you're losing because of this destructive interference. So anyway, as a consequence, you need to design your substrate properly if you want to get an optimized signal. And this is what we have been trying to do recently, trying to obtain distributions of small clusters of gold particles so that we would obtain active hotspots in between each of the particles, which may even be enhanced compared to a random organization of particles. So we teamed up with Agustín Mí at the Institute of Material Science in Barcelona, who has been working on the fabrication of polydimethyl siloxane substrates with little holes that we can now use as a stamp to direct the organization of gold nanoparticles into hierarchically organized assemblies of these particles. So what you see here are SEM images of something that looks like this. So each of the dots that you see here, very well organized, very close to each other, is something like this. So it's made of a small number of gold particles which are organized in a crystalline superlattice. So we have at least 3 levels of organization in this system. And this is important because the separation distance between these clusters within the 2D array is of the order of the wavelength of light. And so you can actually get also light diffraction. And then you obtain something that we call lattice plasmon modes that are characterized by these very sharp bands, which you can tune by tuning the separation distance between the clusters of nanoparticles in the 2D array. So you're tuning diffraction with plasmonic enhancement, so you get an extra degree of enhancement of the electric field at the hotspots within each of the clusters. And this we have already started to use to try to monitor metabolic processes which are characteristic of cancer cells. So for example, here we were looking at the conversion of tryptophan into kynurenine, which is known to be catalyzed by an enzyme that is called IDO1, which is typically overexpressed in many types of cancer cells. And so the first thing we measured, or we compared the surface-enhanced Raman spectrum with the Raman scattering of the pure molecule for each case. And then we find peaks that can be clearly distinguished between the 2 metabolites, so that now we can incubate cells, extract the supernatant over time, and then see how the peak of tryptophan is decreasing over time, while the peak of kynurenine is increasing over time. In the same initial manuscript, we also described an analysis of cell death. So we were incubating cells, submitting them to stress so that they will die, and then measuring what is excreted from the cells when they are dying. And so my student Javier Plo, who has been an excellent driver of this project, designed this cancer on a chip model, which is basically a reservoir where you can incubate the cells, and then you can use one of our plasmonic substrates as the window on top. So you can actually measure directly the supernatant by illuminating from the top and collecting the Raman signal also on the top. And so you can do directly the measurements. You see the conversion of tryptophan into kynurenine, but you also see the formation of this other peak at a bit lower wave numbers, which we assigned to these purine derivatives that are secreted by the cells when they are dying. And then you can do this over time as well, so you can watch the evolution of the cells as they are dying. Now, I guess some of you may be thinking, well, this is very wishful thinking, because there are lots of peaks that you don't even look at, and you're simply focusing on 2 or 3 peaks to derive all this information about the complex biological system. And this is true, and this is why I cannot tell you very much. We are still working on it, trying to write a manuscript, but we are really starting to use more sophisticated data analysis to monitor and even to distinguish different cell death mechanisms. So we started by using the standard PCA, principal component analysis, where by analyzing the full spectra for different stimuli that would induce cell death, we can end up separating the different populations and actually assigning a type of or a cell death mechanism to a certain sample, depending on how you stimulate this cell death. Even more complex, we can also use machine learning, where you have to obtain a large amount of data to train your system, and then you establish connections, and you end up selecting a number of different components that you can correlate with each other, so you actually get a fingerprint that then you can use to identify the mechanism underlying cell death in a certain system. So this is something that I will explain more maybe in the next shift conference. Now, you need a lot of data, and so this way of preparing the measurements is not very convenient, because you have to lift the substrate every time, and then come with another of these plasmonic substrates and put it again carefully on top of your cell culture, so that you can measure with the certainty that you will see what is happening at a certain time. And in fact, if you don't do that, you may end up with what I mentioned earlier, the memory effects. So this is an example where one of these superlattice arrays of gold nanoparticles was incubated with mercaptobenzoic acid. So you were seeing this very well-defined peak that is characteristic at this wave number region. And then you wash the substrate, and then you incubate it in a solution of nicotinamide, and then, surprise, surprise, you only see the peak of MBA that was already present on your particles and not desorbing. And you can also incubate in a mixture of MBA and nicotinamide. You still only see the peak of MBA, and this is what we call a memory effect. And this is why you need a new substrate every time. So in order to be able to collect many measurements and not waste lots of gold and lots of time in preparing substrates and replacing them all the time, my students Javier and Matthias actually designed a system that allows you to recycle your substrate many times. And this comprises spin coating a thin layer of PLGA, polylactic glycolic acid, on top of the substrate. So now actually this picture is not bad quality. It's simply in SEM, you see the projection through the polymer layer, and this is why the plasmonic clusters are seen blurred in the image. This means that the substrate is protected. So if you now measure at any spot here, you will not get any signal. But what you can do is, with the same laser that you typically use for measurements, you can increase the power, and then because of the plasmonic heating effect that you can see here, you see this bright spot is what you see by using the thermal camera, meaning that precisely at this spot the laser is heating significantly. You can actually degrade the PLGA and open a window. And now you have the particles, you can see them again very well, and now your analyte can come and deposit on the particles, so you only measure at this spot. And then you can go and move to a different spot, increase the laser power again, open another window, incubate with a different analyte, and you record a different spectrum. And you can do this as many times as you can shift the position of your substrate and drill a hole. We calculated for a standard substrate that we use, we could do a few thousands of measurements with the same substrate without any removal and redeposition. And so we can do exactly the same thing for the biochip that I mentioned earlier. So now we have this system here with the Plamoni substrate as the window, and then we don't touch it, we simply shift it a little bit, and then open another window, and we do a measurement. So if you compare now what you get by using this trick of opening holes at 0 hours and 24 hours with taking supernatant at the same times, you see that the result is essentially the same. So now we are in a good position to do really lots of measurements over time, also to train our machine learning system. All right, now I switch to the 3D aspect of the project. So the reason that we want to work in 3D is that standard 2D cell culture are not really reproducing what happens in your body, because the cells are attaching to a solid substrate. They almost don't see each other. There is no extracellular environment with all the components that you have in a typical tissue. And so if you don't want to go directly to patients or patient-derived samples, you can be somewhere in between and prepare a scaffold in 3D that allows you to culture something that looks like the real tissue. And so among the different possibilities to do this, and taking into account the ratio between cost and relevance to reproducing a real system, we decided to work with, oops, I went a bit too fast, but that doesn't matter. We decided to work with 3D bioprinting. And this is a mechanical system with an extruder where you can simply insert an ink that is pre-designed and pre-characterized so that it will be fluid when it goes through the nozzle, but will solidify as soon as it is deposited on the substrate. And so you have to do a lot of rheology to understand typically the shear thinning effect of your inks. And then you can create, in principle, samples like this. And so the composition that we chose, we actually have a variety of ink formulations now, but they all contain some polymer and also gold nanoparticles, which can be nanorods, it can be nanostars, depending on how you want to do your sensing afterwards. And so the idea is that now by moving this nozzle in 2D and in 3D, you can obtain these three-dimensional systems that can be cross-linked and where you should be able to detect metabolites and also to grow the cells that you want to have in your tissue. So these are some of the initial images that we got. So you see we reproduce perfectly what was in the drawing. We have this network of holes, which is defined by this hydrogel that if you look by using an ultramicrotome in the electron microscope, you see that there are, in this case, gold nanorods well distributed through the full network of your hydrogel in the scaffold. So now we are in a good position to start thinking about detecting metabolites. And so this is what you can do. This is a very simple example of detecting adenosine. So you can see this peak, which is characteristic of this molecule, of this metabolite. This is the signal that you get with clean water. Here, this is not so intense if you do the measurement in cell culture media, but you still see the peak, which is defining the presence of the adenosine. So now if you focus on this wave number, you can actually do tomography or confocal microscopy so that you actually get an image of the same scaffold, but based on surface-enhanced Raman scattering signal of adenosine, meaning that this analyte is infiltrating through the hydrogel everywhere, and also that you have gold nanorods everywhere, because this signal only comes from those areas where adenosine molecules are binding gold nanorods, okay? So this means that the design of the system is working well. Now, the third part, I told you about encoding particles and using them to label cells and watching them in 3D. So this is a system that is known already for quite some time. Michael Nathan was probably the first one to work on something like this back in the 90s. And so we have simply optimized this with the morphologies that now we can make, which are more efficient as individual particles, typically nanorods or nanostars. So you fabricate the colloidal nanoparticles, you attach what we call Raman reporters, which will be those that will define the signal that will encode your nanoparticle, and then you cover them with a protective layer that will, first of all, avoid leaching of the code, and second, will avoid other molecules to come in contact with your metal particle and contaminate the signal. And then you can also attach antibodies or other biorecognition elements on the outer surface if you want your labeling to be highly selective. And so this is an example that came up from my lab. Dorleta Jimenez de Ballesturi designed this system where you have gold nanostars coated by aromatic thiols that will provide a strong and well-defined Raman scattering signal, and then a block copolymer that will surround it and will make it hydrophilic again, so that you can disperse these particles in ecosystems, also in biofluids. And so this is an example where the nanoparticles have infiltrated cells, and these are the peaks that you can measure directly from imaging the cells. And because of the enhancement from the nanostars, the signal of your label will be much higher than anything that will scatter Raman around it. The advantage of this system is as well that you can select many different aromatic molecules which will provide you with different fingerprints. So you can have almost an unlimited number of codes that you can incorporate onto your particles. And then you can label cells specifically. So the particles will go into the endosomes and internalize the cells, and then you can watch a distribution of cells over time, over long periods of time, because these codes do not bleach. So this is one of the main advantages compared to fluorescent labeling. They don't bleach, and the peaks are very narrow, and this is why you can expand the library of codes. So now we can do this also in 3D. This was probably the first paper showing this, that you can have different layers of different cell types encoded with different nanoparticles, and then you can distinguish perfectly how the layers are growing on top of each other, sometimes interdigitating with each other. If you want to read more about this, there are some recent papers where we describe also monitoring over time and even monitoring cell division over time. So, if you now incubate with cells, the same scaffolds I showed you earlier, then by exciting with the same laser, we are typically using a 785 laser, which is not damaging for the cells, but can excite the Raman scattering of any of the molecules that you want to detect. You can measure at the same time the signal of adenosine on the scaffold. You see the green labels here, and the codes on the cells, which would be the red areas that you see around here. So this again was one of the first examples where we showed one cell type with one code growing inside of this scaffold, which was encoded by a different molecule. And so, as I say, we have a few new papers describing more advanced measurements on this with these systems. I just wanted to show you one more example here related to the advantages of these type of systems. So, as I mentioned, you can encode the nanoparticles with different Raman labels. But if you need, they don't have to be necessarily innocent labels. You can also use useful labels. For example, if you use mercaptobenzoic acid as your Raman label, the signal that you obtain will depend on pH, meaning that now you can also detect pH changes by watching the changes in the spectral signal of these particles. So maybe not so clear here, but you have this peak which remains basically constant in all measurements, but then here you see that as you increase pH, there is this second peak here and another here, which are growing basically at a threshold around 5.6. And so you, by measuring the ratio between the intensity of this peak and the intensity of that peak, you can decide which is the pH around your nanoparticles. So you can also run calibration curves. You can obtain a calibration line from where you can, in principle, extract from the normalized intensity ratio, from the intensity ratio between the two peaks that I mentioned. You can decide which will be the pH that you have around. And this is a correlation between what we measure with these nanoparticles and what we measure with a pH meter, where the correlation is actually working very well. And you can run it also multiple times because, of course, the protonation, deprotonation of this system is fully reversible. And now you can actually internalize the particles inside of cells. And here, actually, we did a further design in this system by depositing an extra layer of, what was it? Polyarginine, I think it was, which is positively charged. And positively charged nanoparticles internalize into cells much more easily than negatively charged. So this is a comparison of what you get with same particles coated with polyarginine and coated with polyethylene glycol. So with polyethylene glycol, they almost don't enter. You don't get any signal from the cells. By coating with the positive layer, you get internalization in all the cells floating around. So you get a very well-defined signal from your mercaptobenzoic acid. And now you can even watch what happens over time. So what we do is we take the cell culture, we incubate with the nanoparticles, we let it internalize for a couple of hours, and then we wash, and then we measure again over time, and we can monitor how the signal changes. So here, I don't know if you can see properly, there are two cells that can be seen in the bright field image. And now you can monitor either through the intensity or even through the corresponding pH value. You can see how the signal goes over time from being mostly around, so on the cell membrane, to being inside close to the nuclei. And you can watch this also in confocal mode, so you can actually see that at different depths, initially you only see the signal around, and after 8 hours you see it mostly inside. And you can correlate this with pH value. So typically what you see is that when the particles are not yet internalized, they typically have pH values above 6. When they are fully internalized, they are all at lower pH because they have been entered into the endosomes, and as they mature, the pH is going lower. So you can really monitor all these processes by simply looking at your encoded particles. Carlos gave a lecture earlier this week showing the first data related to incorporating nanothermometers also in these 3D scaffolds. So by, again, using rare earth doped nanoparticles, you can obtain this calibration curve of intensity or intensity ratio versus temperature. And you can also recover the 3D image of the scaffold by looking at the fluorescence from your nanothermometers. And this is a correlation between the temperature that we measure from the thermal camera to the temperature that we measure from the nanothermometers here. So this is coming up. We are going now to, the objective is incorporating all these different signaling systems so that we can monitor different processes in the cells. And actually, I want to show you a small example of how these systems can be used to test anti-cancer drugs. So again, the same type of scaffolds, maybe a little bit different formulation, the same gold nanorods. And then a slightly different chip where we can deposit our 3D scaffold. And we have here a reservoir where we can inject the drug that we want to test. So the idea is that as soon as you inject here, the drug will start diffusing through the system and will be recorded through the nanoparticles on the scaffold. and we can correlate that with cell death. And so we did this experiment initially with methylene blue, which is actually a photoactivated anti-cancer drug. So it only works to kill the cancer cells after you irradiate with red light. So again, we can obtain a very well-defined signal, and then we can culture, in this case, I think it was HeLa cells inside of the holes in between the lines of your scaffold. So all the green dots that you see here come from the green fluorescent protein that is expressed in these cells. And it shows that there is a very uniform distribution of cells, maybe forming also some aggregates. And then if you inject the methylene blue and activate, if you measure only with the gel on which the cells will grow, you see diffusion slowly over time because this is a viscous medium. So you see that over time, the area where methylene blue has been diffusing is increasing. After 24 hours, it has diffused over the full system. If you incorporate the cells, the diffusion rate is actually quite similar, but the intensity is lower. And the reason it is lower is that part of this methylene blue is internalized by the cells, and so it can be used to kill the cells. And this is actually what you see here. If you look at the control, the green dots would signal alive cells, the red dots would signal dead cells. So in the control, a few cells are dying, as usual. After injecting methylene blue and activating, we see a lot of cell death that is also decreasing along the extension of the system. So this is something that we are now trying to extend to a bit more complex cellular systems and also to more relevant anti-cancer drugs. Now, if I have 5 more minutes, or maybe 3, yes, I'm gonna switch gears completely. Not completely, still plasmonic. But I'm gonna try to tell you a little story about biomedical applications of plasmonic chirality. And this is something that lies close to Nathario's heart. You all know about chirality in molecular systems, which can come either from chiral carbon centers, or it can also come from supramolecular effects, so from the organization of molecules, even achiral molecules, which may end up forming a chiral supramolecular system, right? So it has been predicted that even though standard metal nanoparticles will not be chiral because they are highly symmetric in principle, they should be possible to fabricate this type of particles that have morphological asymmetry or dissymmetry. And this was first demonstrated by using lithography methods with which they can basically sculpt anything on a surface. These are, I have to say for the physicists, these are tedious methods, very expensive, so don't use them. And so the challenge is, can we do this using colloid chemistry? So of course the answer is yes. I'm not going to talk very much about this, but these are examples that we have recently reported. By using different methods, we can grow either tiny grooves that are roughly helical or more extended, twisted nanorods that display also plasmonic chirality in circular dichroism. So you can actually fabricate the different enantiomers, and you can obtain the mirror CD signal from almost enantiomerically pure chiral nanoparticles. But this is not the story for today. For today, I wanted to tell you about the supramolecular chirality. So this is something that also has been done for quite some time, typically using DNA or DNA origami as a scaffold, where you can selectively decide where there will be functional groups that will allow you to attach nanoparticles exactly at the spot that will end up forming a helix or simply a staircase of gold nanorods with different orientations. And by doing that, you can also obtain well-defined plasmonic chirality. Now, a bit more than 10 years ago, we showed that you can do the same thing with gold nanorods. And because of that, we applied for a grant for a Marie Curie fellowship to demonstrate that by using this effect of collective plasmonic chirality, you should be able to detect the formation of amyloid fibers, which are relevant for many neurodegenerative disorders. And so the idea, as you probably know, is that these amyloid proteins, when they get scrambled for some reason, they start to aggregate into small species, oligomers, protofibrils, and then very extended and rigid fibers, which are known to accumulate in some parts of the brain and mainly in what is called the substantia nigra. And it's still unclear. Actually, there has been a quite big scandal recently regarding what is the actual damaging species in this process. So there seemed to be a clear demonstration that it was the protofibrils which were the most damaging ones, but apparently there was a lot of fraud in the publications. So anyway, we don't know very well, but we know that these are very likely responsible for the brain damage. And so the idea now is that because we know that some of these fibers are also helical or double helical, we would like to deposit nanorods and detect the formation of the fibers through the CD signal in the visible or in the near infrared. So they are definitely not coming from biomolecules floating around, okay? And so we started working with a model protein, alpha-synuclein, which can be expressed from bacteria, and we were lucky to collaborate with Archiver Cortajarena in our institute, who can actually express it and then incubate following the literature to actually scramble the protein and generate the formation of the fibers in the lab. So we didn't have access to the top-notch cryo-electron tomography systems, but we trusted that these fibers that we saw in the standard TEM would correspond to the double helices that we had seen in the literature. And so now, if you can do this, you can also incubate with nanorods. And so if you're incubating your fibers and then extract samples of different types and mix them with your gold nanorods, you kind of see these CD signals that are increasing intensity over time. And if you plot the intensity of the CD, you obtain a sigmoidal curve, which is typical of protein aggregation processes. Now, of course, we have to confirm this, so we went again into the TEM for watching at samples at different times. And we were happy to see that there were these lines of nanorods getting more and more extended over time. And also because I'm by nature optimistic, I wanted to see here some places where the rods were crossing each other, so showing a double helix. But also because I'm a critical scientist, and more critical with myself than with anybody else, I wanted to make sure that this was reproducing what we were expecting. And then you resource to good friends. So we have here a common friend, Professor Sarah Bals in University of Antwerp, who can do excellent electron tomography. And so she provided movies like this that clearly show the double helical organization of the nanorods, which of course Jorge had already seen. So, you know, here you have the helix and the nanorods, right? All right, so now we know that this works, and now we have to show that it can be used in real systems. So we first asked Nazario if he wanted to give us a piece of his brain, but he didn't want to. So we had to go to the biobank and obtain brains from dead people, 2 from people who died from something else, 2 from people who died from Parkinson's. And we know that these amyloid fibers are characteristic in Parkinson's. And then by extracting the substantia nigra, and this is what our colleagues, Joaquin Castilla and his group did, and then preparing a homogenate and incubating with nanorods, we could actually see that the Parkinson's brains would give the CD signal very well defined, while the control brains would not really give anything above the noise level. So this is still a bit preliminary. We keep working on this, but it shows that this plasmonic system can actually be useful to at least learn how these systems work. So in the end, the final target would be contributing to the transition between the one-treatment-fits-all medicine to the personalized diagnosis and treatment that we are trying to achieve in the future. So try to simplify the tests that we need to stratify patients so that perhaps in the future we can include also the SIRS test or the CD test in the clinic. With this, I finish. I'd like to thank funding agencies, many bright students and postdocs, current and previous in my lab, And of course, thank you for your kind attention.
Thank you, Luis, for this encouraging presentation and fantastic presentation with so unlimited applications, apparently. So the time is open for discussion. Is there any question on the audience? Please? Come on. The party finished yesterday, so please.
Conference dinner effect.
Thanks, Professor, for the very nice talk.
Thank you.
My name is Fernando. I'm from Unicamp in Brazil. And, well, just a curiosity, I saw in your CD spectra that you have a bi-signature. And sometimes you have positive and sometimes a negative. That you are controlling by micrography or something like that?
Right. In the, say, collective chirality, we are really not defining what the enantioselectivity will be or the enantiomeric signal will be, because this is designed by the scaffold. So since we are using sort of natural scaffolds, they will all have similar handedness, but we don't know which one it is. In the case of the single particle plasmonic chirality, we do define this by the enantiomer that we use to transfer chirality during the growth of the particles. So you can use, for example, amino acids or chiral cosurfactants to drive the chirality during the CD growth. And so depending on which is the enantiomer that you use in the synthesis, you will obtain one or the other handedness in the final particles.
Related with this question, you got the enantiomers because there is, you couldn't get the enantiomers because there is a chirality transfer from the substrate to the nanoparticle. But by using other nanoparticles, you have also chiral. You would be expecting to have that as the
Yes. So we have colleagues who say that almost every nanoparticle is chiral because they are not perfect. And in fact, if you measure CD for single particles and you do it carefully enough, very often you obtain a CD signal. What is very difficult is to obtain a collective CD signal from trillions of particles in dispersion. And there you really need a high enantiomeric excess.
That was precisely my question. So as you said, when you have not perfect nanoparticles, you're going to have probably CD signals that are not coming from chirality, but from some anisotropy.
Sure.
My question was going to be, is there any way to really know that you are getting a true chiral signal?
Yes, so you have to realize that these measurements are done in solution. So there is no alignment on any substrate, there are no orientation effects apart from what comes from the particles themselves. And they are floating around, so you have signal from every different orientation. So we are really truly convinced that this is true CD. There was another question here.
Thank you. Beautiful talk, Luis.
Thank you.
So in your early the presentation, you mentioned this plasmonic nanoarrays for extracellular biomarker detection. So remember, because I was curious, because this is extracellular detection, but there's certain period of time is blackout, because for certain, for example, enzymatic reaction, there's a time delay. So that means the biomarkers inside the cell then come out of the membranes. So I was wondering if you can combine intracellular and extracellular detection. So you have in situ detection.
Yes.
So maybe inside cell use different type of, you show that this, I think at the later stage of your presentation, you show this particle internalization, show the detection inside cell. I was just wondering if you can combine this, because I saw this chip-based in situ detection. But the only concern I have is that there's certain time you're not really tracing the event. Yeah.
You're totally right. So we have to trust that during this metabolic conversion, there is intake and release of metabolites through the membrane of the cell. And it would be great if we can do the measurements also inside. Yeah, this is also in the plan, and I think many people are working on this. But there are several drawbacks or obstacles to achieve it. First of all, as I showed in the pH measurements, what you're sensing is what is inside of the endosomes. And so you have an extra vesicle surrounding the nanoparticles typically, and it's not yet obvious how to disrupt those vesicles so that the particles will be really in contact with what is floating around in the cytosol, so that you can actually do detection of what is there. So if the metabolites would also enter through the liposome, so to say, membrane of the intracellular vesicles, then we might be able to sense, but it's a bit complicated. But you're right, in order to have a full picture, we should correlate intracellular and extracellular signals.
So, Luis, regarding the chirality of the system you make, I guess you have already thought that, like what happened in zeolites, the use of molecular templates to try to have more defined nanoparticles is also one of the strategies that people is following. But I don't see many papers at all where they can actually get a defined structure for a nanoparticle with template.
Yes, of course, the use of porous systems as templates has been around for quite some time. We actually have a few papers, not only where we show the position of porous systems or porous shells around nanoparticles, but even the growth following the pores as templates, but typically using mesoporous silica. For zeolites, I think, and you correct me if I'm wrong, the pores are too small to really allow for a plasmonic system to grow. And so I know some works where people have grown little clusters which can be fluorescent, even made of metals. But to obtain metallic plasmonic systems, you need a thickness or a dimension which is above at least 3 nanometers. And so, yeah, the number of systems where you can get well-defined porosity with that dimension is not so broad.
I guess, Luis, I guess that encoding nanoparticles, for instance, for image, as you mentioned, but you can do it at will.
You can?
You can prepare these nanoparticles at will, depending on the... And how difficult or how complex is to prepare these nanoparticles?
The growth of nanostars is extremely simple. To preserve the stability of the nanostars is a bit more complicated, but you can learn it easily. And then the encoding process is simply ligand exchange. So you mix the nanoparticles, you have to transfer them typically to an organic solvent if you want to have these aromatic thiols, and then they will bind to the surface of the particles. And then, yeah, so there are reported protocols that any chemist should be able to follow, really. And we can also make rather big batches. So especially for nanostars, the synthesis is robust enough that you can really upscale it very much.
So this is wonderful, so versatile in order to go to...
We actually have, and we still have to report that, but we have a continuous flow reactor that we can also use to make production for larger scale.
Great. Any other question? Please don't miss your last opportunity for asking to Luis.
If not, please join to me to thank you
Thank you.
again, Luis, for a wonderful presentation.